Hello and welcome to my channel. In this video we will be looking at biological explanations of obsessive-compulsive disorder, OCD, which you need to know for AQA A-level psychology in the subtopic of psychopathology. I hope you enjoy this video and find it helpful. Let's get started. There are two biological explanations of OCD, genetic and neural explanations. The genetic explanation. The genetic explanation of OCD suggests that OCD is a genetically inherited predisposition and vulnerability that it passed down from generation to generation through particular and specific genes. SERT and COMT genes have been found to appear mutated in individuals with OCD. SERT, serotonin transporter, gene regulates serotonin levels in the brain. A mutation causes an increase in transporter proteins at a neuron's membrane, leading to an increase in the reuptake of serotonin in the neuron, which decreases the level of serotonin in the synapse and causes mood regulation and problems with emotional processing. Variations in this gene, such as the 5-HTT-LPR polymorphism, have been linked to an increased risk of OCD. COMT, catechol-O-methyltransferase, gene regulates the function of dopamine. A mutation causes a decrease in COMT activity and can affect the breakdown of dopamine, which may influence various cognitive and emotional processes. Specifically, the Val158-MET polymorphism variant has been associated with differences in executive function and susceptibility OCD. Taylor, 2013, identified 230 candidate genes that are implicated in a possible cause of OCD, suggesting OCD is polygenic, a trait or disorder that is influenced by multiple genes, rather than being determined by a single gene. OCD is also suggested to have etiological heterogeneity, meaning that different combinations of genes cause the disorder in different people. The different combinations of genes may also account for different types of OCD. Nested et al., 2010, aimed to investigate whether there were genetic and environmental contributions to the risk of developing obsessive-compulsive disorder, OCD. They conducted a family study using participants with a diagnosis of obsessive-compulsive disorder, OCD, and their first-degree relatives. They conducted structured interviews to assess the presence of OCD and related psychiatric disorders among the participants and their family members. The study also included a control group of individuals without OCD to compare the prevalence of the disorder in families. By analyzing the data collected from these interviews, the researchers aimed to calculate the heritability of OCD and examine the familial aggregation of the disorder. This approach allowed them to determine the genetic and environmental contributions to the risk of developing OCD. They found that identical twins showed a concordance rate of 68% for OCD compared to 32% in fraternal twins. They also found that first-degree relatives have a 11.7% chance of being diagnosed with OCD compared to the general population, where the chance is only a 2.7% chance. They concluded that OCD has a substantial genetic component, with heritability estimates suggesting that genetics account for a significant portion of the risk for developing the disorder. Groothuis et al., 2005, conducted a meta-analysis review of over 70 years of twin studies into OCD using various methods. One of these methods compared the concordance rates of OCD between identical twins and between non-identical twins. The researchers found it was far more likely for both identical twins to have OCD than for both non-identical twins to have OCD. They concluded that genetics does play a role in OCD. Identical twins have identical genetics, and so if there is a genetic component to OCD, it would make sense that both twins would be similarly prone to developing OCD. With non-identical twins, the different genes could explain why one twin was more likely to develop OCD than the other. This meta-analysis concluded that OCD did appear to have a strong genetic basis, with genetic influence ranging from 45 to 65% within children. In adults, it was estimated that genetic influence ranges from 27 to 47%. Carey and Gottesman, 1981, aimed to investigate the extent to which genetic factors contribute to the development of psychological traits, specifically focusing on the heritability of intelligence and personality. They conducted a twin study analyzing data particularly focusing on monozygotic, identical, and dizygotic, fraternal, twins. They gathered information on the intelligence and personality traits of these twins through standardized assessments and questionnaires. The researchers compared the similarities between the identical twins, who share nearly all their genes, to the similarities between fraternal twins, who share about 50% of their genes. By calculating the correlation coefficients for the traits in both types of twins, they aimed to estimate the heritability of intelligence and personality. This approach allowed them to assess the relative contributions of genetic and environmental factors to these psychological traits. They found that identical twins showed a concordance rate of 87% for OCD compared to 47% in fraternal twins. They concluded that both genetic and environmental factors play significant roles in shaping intelligence and personality traits. A strength of twin and family studies is that their findings and conclusions suggest that OCD has some genetic basis. Twin and family studies have been found to show higher concordance rates for OCD compared to the general population. Research has shown that identical twins, who share 100% of their genetic material, have higher concordance rates for OCD compared to fraternal twins, who share about 50%. Research has also shown that first-degree relatives of someone with OCD have a higher chance of being diagnosed with OCD than the general population. This is a strength because it suggests that there is a hereditary component to OCD, indicating that genetic factors may significantly contribute to an individual's risk of developing the disorder. The higher concordance rates in identical twins compared to fraternal twins, as well as the increased likelihood of OCD among first-degree relatives, provide compelling evidence for the genetic basis of OCD. This understanding can help guide future research and potential treatment approaches that consider genetic predispositions. A limitation of twin and family studies is that the findings and conclusions may be due to environmental factors. The higher concordance rate found for identical twins may be due to nurture. Identical twins often grow up in the same household and share similar environments, which can make it difficult to separate genetic factors from environmental ones. This shared environment can lead to an overestimation of the heritability of a trait or disorder. As well, as the social learning theory suggests, family members may also observe and imitate each other's behavior through classical and operant conditioning. Alternatively, family members may be more vulnerable to suffering from OCD because of the shared stressful environment of being in a household or around those with OCD. This is a limitation because it suggests that the influence of environmental factors on the development of OCD cannot be overlooked. It indicates that the shared experiences and conditions of family members may play a significant role in the manifestation of the disorder, potentially confounding the interpretation of genetic contributions. A limitation of twin and family studies is that it cannot be concluded from these studies that genes alone determine who develops OCD. The concordance rates in twin and family studies are not 100%, and therefore not all twins share OCD tendencies despite having the exact same genetics. This suggests that genes only do not create a vulnerability and are not a direct cause, as other factors must trigger the disorder. Environmental influences such as stress, trauma, and life experiences could play a critical role in the onset of OCD. This could explain why some people develop the disorder whilst others with similar genetic backgrounds do not develop OCD. This is a limitation because it suggests that the interplay between genetic and environmental factors is crucial in understanding the development of OCD. It highlights that while genetic predisposition may play a role, it is not the sole determinant, and that environmental triggers, such as stress and trauma, can significantly influence whether someone with a genetic vulnerability actually develops the disorder. A limitation of twin and family studies is that they lack population validity. The sample of these studies often has a sample bias. The populations used in twin and family studies may not be representative of the general population. For instance, twins are often selected from specific regions or backgrounds, which can limit the generalizability of the findings. This lack of diversity can lead to skewed results that do not accurately reflect the experiences and characteristics of the broader population. If the sample is predominantly composed of twins from similar socioeconomic statuses, cultures, or geographical areas, the findings may not be applicable to individuals outside of that specific group. Consequently, conclusions drawn from these studies might not hold true for those with different backgrounds or life experiences, which is a significant limitation when trying to understand the multifaceted nature of disorders like OCD. Moreover, the reliance on specific populations can also overlook the influence of various environmental factors that differ across cultures and communities, further complicating the understanding of how genetic and environmental factors interact in the development of OCD. This is a limitation because it suggests that the findings from twin and family studies may not accurately reflect the complexities of OCD in the general population. When studies focus on a narrow sample, they risk overlooking the diverse factors that contribute to the disorder, such as varying environmental influences and different cultural contexts. As a result, the conclusions drawn may be too simplistic and fail to capture the full picture of how OCD manifests in individuals from varied backgrounds. Neural explanations. There are two types of neural, brain and nervous system, explanations: neurochemical and neurophysiological. The biochemical explanation suggests that neurotransmitters, which are responsible for relaying information from one neuron to another, are linked to obsessive thoughts and compulsive behaviors. The neurotransmitter serotonin regulates mood. Low levels of serotonin in the brain have been associated with a disruption to normal transmission of mood-relevant information, as serotonin is removed too quickly before the impulse has passed and transmitted its signal, and therefore mood is affected. Additionally, the neurotransmitter, dopamine, is thought to regulate motivation and concentration. High levels of dopamine have been associated with an ability to focus. There are 4 neurophysiological explanations. One explanation suggests that hyperactivity of the basal ganglia is linked to repetitive motor function, and therefore disturbed communication in this structure might account for repetitive behaviors, compulsions. A second explanation suggests that OCD is linked to abnormality and excessive activity in the orbital frontal cortex, OFC, also called the worry circuit, which is associated with logical thinking, decision-making, impulse translation, and the regulation of primitive aspects of behavior. The caudate nucleus located in the basal ganglia is responsible for suppressing signals from the OFC, and when this is damaged or abnormal, it dysfunctions and fails to suppress minor worry signals, resulting in the primal impulses continuing even after performing the behavior, leading to obsessions and compulsions. A third explanation suggests that OCD is the result of abnormal functioning in the parahippocampal gyrus, which is linked to the regulation and processing of unpleasant emotions. Finally, a fourth explanation suggests that OCD is linked to damage to neural mechanisms due to issues with the immune system, as a result of infections and diseases such as Lyme's disease, leading to the exacerbation and development of OCD. A strength of neural explanations is that there is research to support the idea that particular neurotransmitters cause OCD. Sechtman et al., 1998, found that when animals were given drugs that increased dopamine levels, their behaviors resembled that of obsessive behaviors found in people with OCD. This suggests that high levels of dopamine cause OCD. Piggott et al., 1990, found antidepressant drugs which increase serotonin activity have been found to reduce obsessive tendencies and symptoms in patients. Other types of antidepressants which do not increase serotonin activity have been found to be ineffective. This suggests that low levels of serotonin cause OCD. This is a strength because it suggests that there is empirical evidence linking neurotransmitter levels to the development and treatment of OCD. The findings from studies like those of Sechtman et al. And Piggott et al. Provide a biological basis for understanding OCD, indicating that targeting these neurotransmitters through medication could be an effective treatment strategy. This supports the idea that biological factors play a significant role in the disorder, which can lead to more targeted and potentially successful therapeutic approaches. A strength of neural explanations of OCD is that there is research to support the neurochemical explanation of OCD. Hu et al., 2006, conducted a study involving brain imaging techniques to observe the neural activity of participants diagnosed with OCD compared to a control group. The procedure typically included the following steps. Participants were selected based on specific criteria for OCD diagnosis. They underwent brain scans, such as functional magnetic resonance imaging, fMRI, while engaging in tasks designed to provoke obsessive thoughts or compulsive behaviors. The researchers analyzed the brain activity patterns and compared them between the OCD group and the control group. They also assessed the levels of neurotransmitters involved in OCD to see if there were any significant differences. He found that individuals with OCD exhibited abnormal activity in specific brain regions, particularly the orbitofrontal cortex, anterior cingulate cortex, and striatum. These areas are associated with decision-making, emotion regulation, and habit formation. The study also indicated that there were significant differences in neurotransmitter levels, particularly serotonin and dopamine, between the OCD group and the control group. This is a strength because it suggests that the neurochemical imbalances and abnormal brain activity associated with OCD can be targeted for treatment. By understanding the specific brain regions and neurotransmitters involved, researchers can develop more effective therapies, such as medication that regulates serotonin and dopamine levels, ultimately providing better outcomes for individuals with OCD. This biological perspective reinforces the importance of considering neural mechanisms in the understanding and treatment of the disorder. A strength of neural explanations is that there is brain scan research to support the neurophysiological explanation of OCD. Saxoner and Rauch, 2000, used brain scan research and found overactivity in the orbital frontal cortex to be linked to obsessions and compulsions. This area of the brain is involved in decision-making and regulating emotions, which may explain why individuals with OCD experience intrusive thoughts and feel compelled to perform certain actions. Additionally, another study by Max et al., 1995, found that hyperactivity in the basal ganglia was linked to OCD. The basal ganglia play a crucial role in habit formation and motor control, suggesting that the compulsive behaviors seen in OCD could be related to dysfunction in this brain region. This is a strength because it suggests that the neurobiological factors underlying OCD can be identified and potentially targeted for treatment. The research indicating overactivity in specific brain regions like the orbital frontal cortex and basal ganglia implies that interventions could be developed to modify these neural processes. This approach could lead to more effective therapies, such as medication or neurofeedback, aimed at normalizing the activity in these areas, ultimately helping individuals manage their OCD symptoms more effectively. A strength of neural explanations is that there is research to support the idea that various physical diseases cause OCD. Fallon and Nields, 1994, found that OCD occurs in around 40% of patients who catch Lyme disease, a bacterial infection spread by ticks. Jonko et al., 2018, found similar results and suggest this may be a consequence of the physiological effects of Lyme disease. This connection is significant because it illustrates how infections can potentially trigger or exacerbate OCD symptoms through neurobiological pathways. The inflammation and immune responses associated with Lyme disease may affect brain function and lead to the development of obsessive thoughts and compulsive behaviors. This is a strength because it suggests that the biological underpinnings of OCD may be more complex than previously thought, incorporating not only genetic and neurochemical factors but also the impact of physical health conditions like Lyme disease. Such insights can lead to more targeted therapies that address both the psychological and physiological aspects of the disorder, potentially improving outcomes for those affected. Additionally, it emphasizes the importance of a comprehensive understanding of OCD that includes the influence of infections and immune responses on brain function and behavior. A limitation of neural explanations of OCD is that studies supporting abnormal brain functioning have not been replicated. Aylward et al., 1996, conducted a meta-analysis that included 9 studies published between 1989 and 1995. These authors did not find empirical support for metabolic or perfusion dysfunction in the caudate in OCD across studies that used PET or SPECT. This is a limitation because it suggests that the evidence for neural explanations of OCD may not be robust or conclusive. The failure to replicate findings across multiple studies indicates that the relationship between brain functioning and OCD symptoms may not be as clear-cut as initially thought. This uncertainty can lead to skepticism about the effectiveness of biological treatments and emphasizes the need for further investigation into the neurobiological mechanisms involved in OCD. A limitation of neural explanations of OCD is that a cause and effect relationship cannot be established. Neural explanations of OCD assume a causal link between OCD and neural mechanisms. Neurochemical imbalances and structural abnormalities in the brain may be a symptom OCD rather than the cause. For example, high dopamine levels or low serotonin levels may be the effects of having OCD rather than the cause itself. There could be unknown variables that are not accounted that cause the disorder. This could lead to treatment fallacy, where there is a misconception or misunderstanding about the effectiveness of a treatment for a particular condition. This fallacy can lead to unrealistic expectations about treatment outcomes and may hinder patients from seeking other potentially effective options. From a biological perspective, the best treatment for OCD would be drug therapies, such as SSRIs. However, psychological treatment such as CBT and other talking therapies may be more benefit some individuals. This is a limitation because it suggests that the relationship between neural mechanisms and OCD is not straightforward. It implies that focusing solely on biological treatments may overlook other important factors contributing to the disorder, such as psychological and environmental influences. This narrow perspective can lead to ineffective treatment strategies and prevent individuals from exploring a more comprehensive approach that includes both biological and psychological interventions. Biological evaluation. A strength of the biological explanation of OCD is that there is animal research to support OCD being biological. Amori, 2016, aimed to investigate whether specific alterations and manipulations in the corticostriatothalamocortical (CSTC) circuit are associated with the symptoms of OCD by utilizing genetic and pharmacological methods. Animal, specifically mice, were used to determine how these neurobiological changes contributed to the compulsive behaviors and intrusive thoughts of OCD. He observed the effects of these manipulations on behavior, particularly focusing on compulsive-like behaviors that are analogous to those seen in OCD. Behavioral tests were conducted to assess the frequency and severity of these compulsive actions. He found that alterations in the CSTC circuit were significantly associated with compulsive behaviors in the animal models studied. The research indicated that specific disruptions in this circuit could lead to increased compulsive actions, mirroring the symptoms observed in OCD in humans. This is a strength because it suggests that the biological basis of OCD can be effectively studied and understood through animal models. By demonstrating a link between the CSTC circuit alterations and compulsive behaviors, the research provides valuable insights into the neurobiological mechanisms underlying OCD. This reinforces the idea that biological factors play a significant role in the disorder, which can help inform treatment approaches. However, a limitation of animal studies is that their findings and conclusions can often be generalized to humans with caution. While animal models can provide valuable insights into biological processes and behaviors, there are significant differences between species that can affect the applicability of the results. For instance, the complexity of human cognition, emotions, and social interactions cannot be fully replicated in animals. Additionally, the ways in which animals respond to certain stimuli or treatments may not accurately reflect human responses due to differences in physiology and environmental influences. This is a limitation because it suggests that the findings from animal studies may not be directly applicable to humans, which can lead to incomplete or misleading conclusions about the nature and treatment of disorders like OCD. The differences in brain structure, behavior, and environmental contexts between animals and humans mean that what works in animal models may not necessarily translate to effective therapies or understandings in human populations. A strength of biological explanations of OCD is that they have practical implications. Biological explanations have led to the development of biological treatments to OCD such as gene therapy and drug treatments. These treatments target the neurobiological mechanisms identified in research, allowing for more effective management of symptoms. For instance, selective serotonin reuptake inhibitors, SSRIs, are commonly prescribed to increase serotonin levels in the brain, which can help alleviate obsessive-compulsive symptoms. Additionally, ongoing research into gene therapy holds promise for more personalized and potentially curative approaches, as it aims to correct specific genetic factors that contribute to OCD. This is a strength because it suggests that understanding the neurobiological mechanisms can lead to effective treatment options. By identifying specific biological factors that contribute to OCD, researchers can develop targeted therapies, such as medications that alter neurotransmitter levels or innovative approaches like gene therapy. This focus on biological underpinnings not only enhances our comprehension of the disorder but also translates into practical solutions that can significantly improve the lives of those affected by OCD. A limitation of biological explanations of OCD is that there are other alternative explanations. The diathesis stress model includes both biological and psychological factors and suggests that certain genes leave some people more genetically vulnerable to suffer from a mental disorder, but some environmental stressor or trigger is necessary in order to trigger the condition. The cognitive explanation suggests that the thoughts themselves as the cause of OCD. For example, a cognitive explanation of obsessive-compulsive cleaning may see the sufferer as having persistent irrational thoughts that focus on anxiety-generating stimuli, e.g. a thought that germs are everywhere and going to infect and kill them. Finally, a behavioral explanation would suggest that the reduction in anxiety that is associated with these thoughts, e.g. cleaning, become compulsions through classical and operant conditioning. This is a limitation because it suggests that the biological perspective alone cannot account for the complexity of OCD, as it fails to incorporate the significant roles of environmental triggers and cognitive processes. By acknowledging alternative explanations like the diathesis stress model and cognitive behavioral approaches, it becomes clear that OCD is influenced by a combination of genetic vulnerabilities and learned behaviors, rather than being solely determined by biological factors. This broader understanding allows for more comprehensive treatment options that address both the biological and psychological aspects of the disorder. A limitation of biological explanations of OCD is that it is biologically deterministic. This means that these explanations may oversimplify the complexity of the disorder by attributing it solely to biological factors, such as genetics or neurochemistry, while neglecting the influence of environmental, psychological, and social factors. By focusing exclusively on biological determinants, this perspective risks ignoring the role of personal experiences, upbringing, and cognitive processes that can also contribute to the development and maintenance of OCD. For example, an environmental explanation could suggest that people are not born with OCD and learn the anxiety disorder through the learning process of operant and classical conditioning. This is a limitation because it suggests that OCD is primarily a result of biological factors, which can lead to a narrow understanding of the disorder. By not considering the interplay of environmental, psychological, and social influences, this perspective may overlook important aspects that contribute to the onset and maintenance of OCD. It implies that individuals have little control over their condition, potentially undermining the effectiveness of psychological interventions that address learned behaviors and cognitive processes. A limitation of biological explanations of OCD is that it is biologically reductionistic. This means that these explanations tend to simplify the disorder by reducing it to just biological factors, such as genetic predispositions or neurochemical imbalances, while overlooking the complexity of human behavior and experience. By focusing primarily on the biological aspects, this perspective fails to account for the interplay of psychological, social, and environmental influences that can also play a significant role in the development and manifestation of OCD. For instance, cognitive behavioral factors, such as maladaptive thought patterns or learned behaviors, are crucial in understanding how OCD symptoms develop and persist. Additionally, social context, including stressors and relationships, can greatly impact an individual's experience with OCD. This is a limitation because it suggests that OCD can be fully understood and treated solely through biological means, which may lead to inadequate care for individuals who also need psychological or social support. By ignoring the multifaceted nature of the disorder, this reductionistic view can hinder the development of comprehensive treatment plans that address all contributing factors, ultimately affecting the recovery process for those affected by OCD. Thank you for watching. If you have enjoyed this video, please like and subscribe for more content like this. Bye.